All right, in the interest of time, let me tell you a couple of other things. So one, I'd like to kind of tell you about the general notion of programmable medicine. So if I bucket things we're working on, they fall under a couple of categories. One important category set is programmable medicines for us. And so, in fact, is this, let me just make sure, okay, good. So in this regard, let me just say what I mean by programmable. We've lived in the last 30 years side by side with another exciting industry. It's called tech, digital, that has undergone a dramatic transformation and impact on the planet. And while I'd like to believe that biotechnology has had a great impact, it did in the last couple of years, but more generally, it's been a pretty niche space. And it's always been interesting to watch what's causing tech to grow quite this way. And I would argue part of it is that it's a code-based technology. In other words, you can actually program, you can modularly exchange things, you can do all sorts of things with code that if everything that you produce is a bespoke one-off thing, every time you do a product, it starts from scratch. So it's always been of interest for us, obviously, since the genomics work, sequencing work, that the original IT industry was actually biology. I mean, it's a code industry, right? DNA is the code, mRNA is kind of the software that gets made. All of this has been nice metaphors, but not actionable, until the advent of nucleic acid-based medicines, which I know is a great area of interest here. So let me kind of tell you how we think about it. We think of cells as basically the computer, so effectively what processes the code. And the question is, how do we safely be able to get instructions? Well, gene therapy is one way people do it, gene editing, gene writing. There's a lot of different ways. Our first approach was with mRNA, and let me tell you a little bit about that. So back in 2010, we, within our labs, in an entity called LS18, which was the 18th such company we started asking questions like this. What if a patient could make their own biotherapeutic inside their body? What if that could be transient instead of incorporated into the genome? And what if you could actually do this in a way that once you got good at it, you could do it over and over and over again in a very short time frame. So this notion of programmability. Those set of questions led us to look at it in all sorts of different molecules, of which we felt that as a starting point, mRNA, despite all of its known disadvantages, innate immune response that it causes, relatively labile, hard to get into cells, hard to get into the right cells, unknown whether in an animal it'll make the right protein, folded in the right way, secreted, or at the right level. All of what I just said were uncertainties in 2010. Anybody who estimated the probability of success of mRNA in 2010 would have been mistaking uncertainty for risk. There was just pure uncertainty. But the question was, can you build a platform to actually reduce this to practice? Can you do this long enough until you overcome these challenges? And that's what we set out to do. Today, what exists is a platform that has been applied to many different aspects, and I'll tell you a little bit about this. But it essentially has 48 molecules that came out of the same exact platform that are in various forms of preclinical and clinical development, of which 30 are in human trials. The original idea was this. The original idea was, could we somehow get RNA into cells, somehow avoid innate immune recognition, and then basically make any protein we wanted? And this is kind of the journey that we undertook. Initially, it was about reduction to practice and proving some of these elements could even be done through chemical modifications, through initial delivery approaches. And I'm not going to go through every one of these, but what you'll see, interestingly, is ways in which it takes a village, right? DARPA got involved in some of the work that we did very early on. We did a major partnership with AstraZeneca, without which we wouldn't have been able to afford building out the platform. AstraZeneca did a partnership with $240 million upfront capital to work on up to 40 programs together, all based on mRNA. You can't even think about 4 programs if you don't have a reusable code-based approach. But this was something we thought we could make many, many different approaches to. Then many different corporate partnerships continuing all the way to 2018, where we built a plant, even though we didn't have a product. So in Norwood, Massachusetts, we built a mRNA production plant. And the reason we did that is we wanted to support our cancer vaccine work. And then what you'll find also is that by 2019, we had positive human data in CMV. We had positive human data in HPV. We had a bunch of different initial phase 1 trials going on. And then, of course, we had what I call the platform But the idea for the platform was, can you go from an idea, a new protein you want to get made, all the way to the actual formulated product in an animal or a human arm in a relatively short time frame. And we had pulled all these pieces together. You'll have to take my word for it, but you don't have to take my word for it, because you also know that as a result of that, when the SARS-CoV-2 virus showed up, we got the sequence. 1 day later, we had the code for the mRNA we needed to synthesize. A couple of days later, we had the first instance of the mRNA in LNP. We simultaneously tested it in some animal models, but then we were in human clinical trials in 44 days. That is 44 days from knowing the target offending sequence of spike in particular to actually going into a phase 1 human trial. And that time frame, while impressive, is completely due to the fact that we had a platform that we had spent 10 years developing and $2.5 billion developing. $2.5 billion of private investment, public from the public market standpoint, and corporate investments. That all preceded SARS-CoV-2. And then what ensued was essentially the exact same thing that we had the design for, that we had the dose for. A few days later, from the start time, 9 months later, we could show could be administered to 30,000 people, and showed that the effectiveness and the safety was such that it could be a potential solution. Along the way, a lot of collaboration, yet again. Now, in this case, a lot of government entities got involved. A company called Lonza was very integral to helping us scale in Europe, even while we were scaling in the US, et cetera, et cetera. And I'm happy to say more about this if it's of interest. But what was interesting was the time frame, and again, the time frame which some in the press and some skeptics have viewed as being taking corners. We did a more robust, thorough trial with more people than any kind of impending infectious disease trial has ever been done. Of course, there are other viruses for which vaccines have been developed more slowly and with lots of people, but this was done, it's not like we did it in 3,000 patients or subjects. So how could that happen? How could one go so fast? First and foremost, mRNA is an information molecule. Second, we had done nine phase 1 trials before SARS showed up, actually, in vaccines. Every one of them produced neutralizing antibodies sufficient to neutralize a virus in culture. We had worked on MERS, and this was the connection to Dr. Fauci, the infamous kind of partnership that we had that ended up allowing us to move very quickly, because MERS turns out to be relatively similar when it comes to the spike, and that gave us a lead. We owned our own manufacturing facility for a totally different reason, cancer vaccines. Not only cancer vaccines, personalized cancer vaccines. The manufacturing train we used to make the first 100,000 of these doses, let's say plus or minus, was one that we had previously used to make individualized doses for individualized cancer patients. Except it was highly automated, highly digitized, and we essentially told the computer, think it's a new patient, keep making another one, don't change the sequence, and effectively we started scaling up the same approach, which was very interesting. All right, a couple of other things. This is a chart you should try to keep in mind if you're working in the vaccine space. This is a chart that's showing the discovery of the infectious agent and the time when we had a vaccine for it. In some cases, we don't. Malaria, you'll see we don't have a vaccine for, approved. You see meningitis in 1981, on and on. The green kind of things indicate when a vaccine was developed for it. And then you'll see COVID to the bottom right there. And so the question you have to ask, and by the way, the check marks are the things that Moderna is now working on in various ways. The question is, are we going to go back? Are we going to say, okay, just kidding, it takes 5 years, 10 years to do this, given that you know, especially if there is enough incident conditions, that you can actually get a sequence and get it made and get it expressed in the body using the same exact platform that you've now gone into 2 billion people. So one of the interesting things is, I worked for the first 30 years of my career, or whatever, 25 years, on proteins. And then all along, when we started working on mRNA, I used to think, well, geez, this is going to take, I'll be long dead before mRNA enters 10,000 people. Forget about. And all of a sudden, this field has gone from actually being a second also ran to proteins. There's more humans on the planet that have received mRNA and actually reacted through their immune system than there is proteins that have entered humans ever. And so from a interesting kind of balancing of risk-reward, we have quite a lot of data, and we're studying that data to be able to make better and better formulations, better and better approaches. So that's very interesting. So we started with Moderna, but then the idea of programmable bioplatforms has proliferated, such that we're working on such platforms using DNA, using proteins that are now much more programmable, et cetera. And I'm going to give you a couple of quick examples of that. Let me see how we're doing on time. I think we have another 15 minutes. There's 4 examples that I can give you. I'm going to really go through this quickly, and you'll have to forgive me, but I'm happy to say more in our Q&A section. They're all here. They're all different variations on the same theme. Tessera is a gene writer approach. It's attempting to insert whole genes or large chunks of genetic material very precisely, code specifically, into the genome. Alterna is using tRNA as a way to do read-through of premature stop codons, engineered tRNA as the drug. So it's not mRNA, but it's delivered through LNP again. Omega is trying to do epigenomic modifications in order to turn on or off sets of genes through the modifications that allow a whole region to open up or not. So it's not gene editing, it's epigenomic change. And Laurent is developing a translatable version of circular RNA that we think could have some interesting applications. So let me tell you about these very briefly. Again, forgive me for skating through this. The question we asked here is, what if nature has already invented DNA writing tools and we don't have to kind of go and engineer these? Certainly we did that with CRISPR. CRISPR was a nature-based approach. The question is, where in a mammalian system might this exist? And of course, one of the places the team went to look, they looked at lots of other places, is essentially what allows for mobile genetic elements. And when you look at the relative abundance of genes that are involved essentially in allowing for mobile genetic elements, it's gigantic. It's part of the most abundant set of genes that exist. But we haven't really put them to use as a writing technology. And so essentially what the team set out to do is to look at the huge parts list that exists at a genomic level, a sequence level, and start thinking about what about those correlate with the functions that we want. And using machine learning, started learning from those large set of engineered parts that we already have, which ones might be useful as a potential therapeutic. The team set out to work on what we call rewriters. So essentially nucleotide substitutions, insertions, etc. Bigger chunks, exon writers, and then ultimately whole gene writing. Again, very, very specific to a particular sequence. Anybody who's interested in this, Tessera is quite kind of rapidly advancing. There's a lot of papers and patents that have been filed. And it's a team of about 250, 300 people in Cambridge, and they're working on a number of rare diseases and now increasingly cancer. Second case, Omega. Again, super high level. Omega is a public company, so there's even more that's been written about them. Turns out that there are these insulated genomic domains that Rudy Anish and Rick Young and several others in various academic labs have published around that actually show that through these CTCF binding sites, essentially a loop can form that could open or close depending on the epigenetic state of those appropriate bases. By designing what we call an epigenomic controller, and that's what's laid out here, so essentially a DNA binding domain and an epigenomic effector, formulating it into an LNP and getting it into liver at first, and then lung is what we're working on next. What we've set out to do in this program is to actually, for example, go after c-Myc. We're in the clinic, human trials with c-Myc, where we have shown, certainly in animals, a dramatic reduction in c-Myc expression, which we think could be, certainly in liver cancers, quite applicable as well as in lung cancer. But if you can show this can work for 1 or 2 of these targets, we think you can show it for many, many other ones. Again, it has this programmability aspect because you could change the code. You may or may not want to change the effector, and you'll have a different effect. Third one speaks to Laurent I mentioned. So as you know, linear mRNA has the advantage of being translatable, pretty labile, few hour half-life, if that. We also know that there's massive amount of non-coding RNA in the form of circular RNA, not translatable, but actually quite a bit longer half-lives, in some cases days. And so the question we try to ask ourselves is, could we actually use this circular form, which exists in nature, and make it translatable? Not what a lot of other people have done, which is to take the linear form and circularize it as a message in and of itself. So we wanted to get away from the cap and the tail, etc., but to actually work and engineer these. And this is a long-term program that we've undertaken. We continue to work quite a bit on this. This is really heavy lifting in terms of platform creation. People have kind of gotten into the space and have some interesting new approaches. In our hands, there's a lot of platform building to do, and we continue to do that. And it's just laying out. So we're using irises, for which there's very little optimization that's been done. We're doing that in a very, very data-centric way. Actually optimizing the sequence of the ORFs that you're using, the delivery, there are many different approaches, not just LNP. There are some things we found through conjugated approaches that can be done, etc. And the idea is to not only be able to design it, but to actually make thousands of these, test them for many different applications, vaccines, multi-antigen delivery, etc., and then the manufacturing. And then the last one I referred to, Alterna, this is actually saying, okay, can we take a synthetic tRNA and introduce it into cells where the genome contains a premature stop codon and actually read through it? Now, anybody who hears it out for the first time will immediately say, what's to stop what is to stop the read-through of the natural or normal stop codon? And we worried about that, obviously, and we did quite a bit of work up front. And people kind of told us, well, it's probably going to just keep going, and you're going to make these monster proteins, and it's going to make lots of additional bases. But actually, when we went into the lab, what we found was that the read-through, by the way, a ton of optimization went into actually figuring out the right sequence, the right modifications of this tRNA. But essentially what we've shown is with relatively little differentiable read-through of the terminal stop codon, we were able to read through with an ARCH-TGA sequence a premature stop codon and essentially make the natural, normal, full-length protein. To do this, we've also had to develop a whole set of new capabilities, analytical capabilities, design capabilities. A lot of machine learning has gone into this. And so what we're trying to do here, which is kind of laid out, is to actually come up with these types of constructs. You could imagine, if you've worked in this field, that there's many other stop codons you could read through, and there's many other amino acids you can put into where the stop codon was. So that means you can go after all sorts of rare diseases. So it turns out that just the TGA stop codon is involved in all these rare diseases and another 2,000 of them, by the way. So if you go look at all the rare diseases, about 10, 15%, sometimes 20% of them are premature stop codon based. And our hope, although right now it's complete fantasy, we'll see how it works in the clinic, our hope is that we can actually develop a single ARCH-TGA tRNA construct that can be used for many different rare diseases as long as people have that particular stop codon. So you can genetically define the subset of the population and give them exactly the same product, whether they have OTC or MMA or GSD. So think about the bucket trials people have done in cancer and think about whether you could do that with rare diseases. Because the notion that somebody's going to pay to do a de novo trial of a new agent for these small rare genetic diseases is pretty inconceivable. Our sense is maybe with one drug we can treat multiple of them, not 100%, but 15, 20% of the patients. That's the goal of this. In the interest of time, I'm going to skip this just to mention that we have also a lot of efforts ongoing within Flagship in what we call preemptive medicine. This is the attempt to get upstream of disease. Maybe I'll just mention this slide. The notion is, what if we could take sick care? And I've heard in the last couple of days a lot of interesting ideas that are emanating from Singapore about how to think about healthcare differently. We very much believe in that. We've been working for a number of years on approaches that might get ahead of disease. So think about this challenge. What if you could pre-treat pre-patients that have pre-diseases so as to delay or deter the disease? Notice I didn't use the word prevent. Prevent to me is in the category of words that go along with cure. I also don't believe in going around saying we can cure. It might happen once in a while, but prevent is pretty high standard. Preempt means that you might get diagnosed with NASH 3 years later, 5 years later. You might get diagnosed with diabetes 2 years later. Well, that's not going to happen on its own, but if you can't catch people within 6, 12, 24 months from diagnosis, we can't try interventions to try to actually delay that. And that's what we're trying to work on. There's some very interesting projects across multiple different companies that are aiming at that. I'll spare you what we're doing in AI machine learning. We'll do it in the Q&A side. And let me kind of end on this slide. And I started with one provocative statement. I'll use a quote in this case from George Bernard Shaw. And just say what I've been trying to tell you is a scientific engineering institutional approach to using imagination and then converting it into action, as opposed to the more traditional approach of iterative kind of adjacencies explorations. And this quote I like, it says, imagination is the beginning of creation. You imagine what you desire, you will what you imagine, and at last you create what you will. Now, he wasn't talking about science, but if we can get ourselves, especially with the help of machine learning, to actually do this type of imagining and reducing to practice, I think we'll have a lot more solutions for the future. Thank you very much.